We are now going to jump into the Exotic Aquatic Plant Watch, and I'm going to let my co-presenter Eric introduce himself first.
I have to remember where the mute button is. Hello, everyone. Thanks for joining us or being with us all day. I am an aquatic ecologist with Michigan State University Extension. I work on lake conservation statewide, and monitoring is one of my big things. And within monitoring, something that I've done a lot of research on and work on are aquatic plants. And so I look forward to discussing the invasive plants out there and our early detection program.
Thanks, Eric. And those of you who haven't been on here already, my name is Jo Latimore. I'm the director of the MiCorps program. I'm housed at Michigan State University in the Department of Fisheries and Wildlife, where I'm a faculty member. And I also serve as the director of our Extension Center for Lakes and Streams. And I won't say we've saved the best for last, but the plant stuff is always a lot of fun for us to talk about. So we're going to dive in. I'm going to give a little background and introduction to the value of aquatic plants and the trouble we have with invasive plants. And then Eric's going to take you through how to identify the top 5 aquatic invasive plants that we focus on in this monitoring parameter. And then we'll end with me giving an overview of the actual monitoring protocol. So aquatic plants are a critical part of our lake ecosystems. Just like the trees and shrubs, that diverse vegetation that makes up a healthy forest, under the water surface, aquatic plants make the really important structure for our lake ecosystems. The more different kinds of aquatic plants we have, typically the healthier a lake is. So it provides habitat for fish and mussels and crayfish and all the little critters you see in that lovely image there on your screen right now. They help improve water quality and much, much more. However, next slide, please. If we introduce plants that aren't supposed to be in our lakes, just like introducing invasive species into a forest cause problems, the same thing happens in our lakes. There are some plants that have gotten into our Michigan lakes that are not supposed to be there. They didn't evolve there. They are not adapted to have predators and things to keep them in check. So when they get into our waterways, they can grow to the point where they cause problems. And that is what we're focusing on in the Exotic Aquatic Plant Watch. The problem with aquatic invasive plants is that they reduce the diversity of plants, because what makes a good invasive species? The ability to grow really fast and outcompete everything around you. And so that's what they do. They reduce diversity by crowding out our native plants that provide good habitat and food for things that live in the lake. They impact our fisheries by diminishing the quality of spawning areas, growing so thick that, little fish can't hide in them and big fish can't hunt in them. They reduce property values by, making it difficult to swim, to boat. They can be unsightly when they grow in big mats on the surface, for example, and they can be very difficult and expensive to control. Next slide, please. So what we first recommend everyone think about is preventing them from getting into our lakes in the first place. And there's a lot of ways to do that, including a couple of programs that MSU Extension offers. Our Clean Boats, Clean Waters program provides information, resources, on-site assistance, and even funding to help communities stop the invasion of non-native species into lakes in Michigan. So I encourage you to check out our Clean Boats, Clean Waters program. We also run our Ripple program, and Ripple focuses on another way that invasive species get into our waterways, not boats, but aquariums and water gardens. Through Ripple, MSU Extension works with retailers retailers and with hobbyists to make sure that people who enjoy the aquarium hobby or water gardening know how to properly dispose or rehome plants and animals that they might have and no longer want. So that's been a very successful program that carries on today. In fact, one of the program areas that Ripple is really focused on right now is invasive crayfish and keeping them out of our waters. So Clean Boats, Clean Waters, Ripple, two different statewide extension programs that can help you. However, prevention doesn't always work. Despite our best intentions, mistakes happen. Hitchhikers get caught on our boats or in our plantings and things, and we although even if we do our best, they may still get where we don't want them to go. I'm showing a couple of headlines here from the infestation of the invasive plant hydrilla in Lake Manitou in northern Indiana. They have been fighting for years. You can see the date on those headlines is 2007. They have been working for years to control the hydrilla infestation in this lake in northern Indiana, which, keep an eye on that, that's not too far from Michigan, has caused them millions of dollars. They've had some success, but they had to take extreme measures to stop this invasion from spreading. And then just recently, as we'll revisit a couple more times throughout this presentation, hydrilla was found established for the first time in a Michigan waterway. In Southwest Michigan, in a private pond, hydrilla was found at the end of last summer. And even though this is a small invasion, we are not taking any chances as a state with this. There is intense effort going on to control the known population of hydrilla there, to look for where it may have spread to, and so forth. So because prevention is not guaranteed, we go to the next step, which is what the Exotic Plant Watch is all about. We're really interested in early detection, and that's where volunteers like you come in. Learning about these invasive plants, what they look like, where you might see them in your lake, and how to report them if you find them. Really, the sooner you detect them, the better, because you could have an invasion like the one on the left. That's a starry stonewort individual there, but it's not covering the entire lake bottom. Unfortunately, it doesn't take long to look like it does on the right, because these invasive species, again, remember, they spread really fast, they grow really thick, and so early detection is critical, because if it's still a small invasion, you may have a chance of controlling it before it gets out of hand. So some of the key features of early detection programs are focusing on the most probable invaders. If we told you about all 120 some species of invasive plants and animals that you might find in your lake, you'll probably run away screaming, think there's no way I'm going to be able to find all of those or look for all of those in my lake. So we focus on the ones that have the highest risk of showing up in your lake. We also focus on the highest risk areas for new invasions, like, as you might guess from that image, boat ramps are areas where we may see invasive species being introduced from trailered boats coming from infested waters. They also require continuous monitoring. You know, every time another trailer comes into the lake, every time someone takes their dog swimming in a pond somewhere and then they run over and jump in your lake, that might bring an invasive species in. There always needs to be people locally available to keep an eye out for anything that doesn't look right, that looks like it's some plant that we haven't seen in the lake before. So if you know what to look for, you'll be better positioned to report and detect an invasive species early. Our program is called the Exotic Aquatic Plant Watch, and it is the state of Michigan's early detection monitoring program for aquatic plants in our lakes. And that's what we're going to be going through today. Once you will learn by participating in this training how to identify the top 5 aquatic invasive plants that we're concerned about that are of highest risk to most Michigan lakes. You'll learn where they're likely to be found and how to report them. So it's, again, as I mentioned before, you want to get involved with the Plant Watch because we know that early detection and rapid response works. And that's exactly what we're doing with hydrilla right now in southern Michigan. Next slide. So the key to understanding our invasive plants is to know those species. And these are the 5 that we're going to talk about today. Eurasian water milfoil, curly leaf pondweed, which is the one shown in the picture there, starry stonewort, hydrilla, and European frogbit. And we have some different resources to help you recognize these species. The one that we rely on most heavily in this program is a Michigan Boater's Guide to Selected Invasive Aquatic Plants. This is available for free download both on the MiCorps site and on the MSU Extension Bookstore link that you see there. If you want hard copies, we provide them to our enrolled lakes, and anyone can purchase them from the MSU bookstore for about $13. They're printed on water-resistant paper and such, spiral bound, so they're a nice resource to have with you on the boat. On our website, we also have a really nice video. I say really nice because I'm in it, so it's got to be pretty good. Now, one of our past technicians did most of the work on putting that together, but it describes the Plant Watch program, gives tips and tricks for identifying the plants. It goes over the sampling protocol. We actually went out in the lake with some volunteers that were in the program and filmed them while they were conducting the program. So if you go to our website under lake training, you'll find that video there, along with videos on a lot of our other parameters as well. So at this point, I'm going to hand it over to Eric to introduce you to some of these exotic species.
All right, thanks, Joe. It's the end of the day. We've been presenting all day, and Tamara had some nice poll questions. We'll just do this in chat. Does anyone know what invasive aquatic plant is in my hand in this picture?
Starry stonewort.
Nice. Good job, Florence. Yep, so that's starry stonewort. We see the classic bulbil here, which is a reproductive structure for this plant slash macroalgae, and it is shaped like a star. So, as Joe mentioned, she mentioned 5 species that we are on the lookout for in the Exotic Aquatic Plant Watch. We split them up into 2 categories. The first one is established aquatic invasive plants. So in Michigan, although maybe we quickly say weeds and we might assume there's a lot of invasive plants that are submersed, there are actually not that many. And so here are the 3 most common aquatic invasive submersed plants in Michigan. They're well established. Eurasian milfoil and curly leaf pondweed has been in the state for many decades. Starry stonewort less than, but still a handful of decades, and has spread to many, many lakes. Our more recent invaders that we asked to look for are hydrilla and European frogbit. As Joe alluded to, but it's good to say again, is hydrilla was found in the state for the very first time in Southwest Michigan near Berrien Springs last year. And so that is being addressed, but as you can imagine, if it's there, it could be in other locations. European frogbit is a more recent invader to our inland waters. And it was in our coastal wetlands with the Great Lakes for quite some time, but then it started to pop inward on inland lakes and ponds and slow-moving streams. So let's get started with some identification. So we're going to step through each of these species now, learning key characters and thinking about some of the lookalikes out there as well. So our first one is Eurasian water milfoil, Myriophyllum spicatum. So how I'll set this up is if you are standing on your boat, you pull a plant up that was submersed in the water, except for maybe a tiny little piece. So submersed in the water, and the leaves look like feathers. So what do I mean by the leaf? And so here we are looking at this right here. This whole thing is the leaf. What we've done is, in this illustration, is cut a node out and are looking at it straight on. So that's the stem, and there's the leaf. So now if we have pulled a plant out, it has feather-like leaves. The next thing we want to ask ourselves is, what is the leaf arrangement? And so leaf arrangement in this case is whorled, and I'll show you what whorled means. So in this node here, there's a leaf coming out in multiple, more than 2 leaves coming out of one node whorled around the stem. I'll show you in our next plant that sometimes it's alternate, where if you can see my tiny video is one leaf would come out here. The next leaf down would come out the opposite side and down of the other leaf. So this would be alternate leaf arrangement. This would be opposite leaf arrangement. And so we have submersed plant, feather-like leaves, whorled leaf arrangement. Then we're going to count the leaflets. And so this right here is a leaflet. And as you can see, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 leaflets on this one here. That means 13 pairs. So you only have to count one side because they are together. So 13 pairs of leaflets. If that's the case, you have Eurasian water milfoil because you are over 12. You have those feather-like leaves and are whorled around the stem. So that's not too bad. Eurasian milfoil can be tricky, but with these simple characters put together, you can get a good glimpse of, or you have the skills to identify it. There are other native milfoils, and I'll get to that here in a moment, which are wonderful and beneficial to the environment. So, for example, we have a native northern water milfoil, or Myriophyllum sibiricum, and that's shown here on the left-hand side. So again, it's a cross-section, that's the stem, and here are the leaves whorled around. But so everything looks very similar, right? It looks like a feather whorled around the stem, but then we have just 1, 2, 3, 4, 5, 6, 7, 8, 9 leaflets. And so it is not Eurasian water milfoil, it is northern water milfoil. Now, these two, unfortunately, have hybridized, and that has made life a lot more difficult to identify. And hybrid milfoil is problematic because it can be very aggressive. And also, some of the strains of hybrid milfoil can actually be resistant to the same types of herbicides that are used for Eurasian milfoil. And so because of that, a good identification is needed. And the only way to 100% identify hybrid milfoil is with genetic testing. And the lab that we are aware of that does genetic testing is with Dr. Ryan Thum. That's Thum. And he is out of Montana State. And he has, on his website, has a form that you can fill out to get your and directions on how to send milfoil fragments to him for testing. But just to show you, here are illustrations of our native, a handful of our native milfoils. So some of our native milfoils have these club-shaped forms on the side. These are winter buds forming. Some of them have really tight whorls, and so they look really fluffy in the water. Some of them, as I mentioned, have under 12 leaflets. And if you can recall, I remember Eurasian milfoil is whorled around the stem, where there is a somewhat common one in at least southern Michigan that is not always whorled. And so you can see down here in the image where it's more scattered on the stem compared to whorled. And so this is whorled milfoil, or Myriophyllum verticillatum, northern milfoil, which is Myriophyllum sibiricum, and this one is variable leaf milfoil at Myriophyllum heterophyllum. There are other characters associated with these, but this is just a quick overview. So milfoils belong in this, when we just are grouping plants together, we call them finely divided leaves. So submersed plants, instead of calling them compound leaves, like we say in the terrestrial world, like an ash tree, we call them finely divided. And so we have a couple groups that we group these plants into, and see these are feather-like, so that's the milfoils. We have heavily branched, but with bladders, and those are called bladderworts. This is actually a carnivorous plant, all native here in the state of Michigan. And these bladders, just if you have any curiosity, type in bladderwort into YouTube, or its scientific name, which is Utricularia, and you can watch in slow motion as these invertebrate comes, triggers the bladder to open, and the invertebrate gets into the bladder and then gets dissolved and digested. And then there's also this one, which coontail, which is a very common plant across the state, and it is commonly confused with our milfoil species. But when you look at just the leaf, so that's what this circle is around, it is like a wishbone or a peace sign. And if you look at that wishbone or peace sign leaf, you can see that there's even teeth on there, where our milfoils do not, and Eurasian milfoil definitely does not. So I don't know, you can call it out or put it into chat. Which plant? So usually if we were in person, we would be able to hold some stuff, but here's the question. Which plant is Eurasian water milfoil? A or B? And pretty interesting, right? Like these are very similar at first glance. Feel free to type it into the chat or even just call it out. Here, let's see.
Got a lot of
All
votes
right.
coming in, almost unanimously for B.
All right. Hooray, Plant Watch team. Thanks, Joe, for that. Yes, so B, and the reason why is because we have everything's very similar, right? A feather-like leaf, world leaf arrangement, but then there are a lot more leaflets on here, 12 plus compared to under 12 on A. So moving on to our next species, which is curly leaf pondweed. There are, I think, 28 pondweeds in Michigan, all of them native but one. And that species is curly leaf pondweed. What's nice is the curly leaf pondweed, just to the punchline, the only curly leaf pondweed is the only pondweed with serrated leaf edges. So when we say leaf margin, that's the edge of the leaf here, and it has serrations. But how do we get there? So if you see a plant in the water, so a submersed plant that's mostly entirely under the water with just potentially the flower coming up above, and then you pull it up and the leaves look like short ribbons, like it's the width 3/8 wide and 2 to 3 inches long. You can easily see a midvein. So what I mean is you can easily see this midvein. Whoops. Then when you look at the leaf arrangement, it's alternate. So alternate leaf arrangement, where you can see it really depicted nicely here. On this leaf is coming up here, the next leaf is coming off on the other side, and then that serrated or toothed edge. There are a lot of lookalikes, but what's nice is there are no teeth on those lookalikes. So here is variable leaf pondweed, can be very similar size, but there tends to be a lot more branching on variable leaf pondweed and no teeth. Another very similar one, a little bit bigger of a leaf, a little wider, is white stem pondweed, which we see here. It even has kind of a ripple effect, kind of like curly leaf pondweed, but there are no teeth, And actually, I'm sorry, I should have gone back to that. These leaves are very much like lasagna noodles. If you look at the edge of a lasagna noodle leaf, it has those ripples. So curly leaf pondweed has that, but not always. So early on in the spring, if your lakes are invaded by curly leaf pondweed, you might go out there right now and they're very flat. But one thing is for certain, if you have curly leaf pondweed, it will always have serrated leaf edges. And so again, here's white stem, where you can't just look at the ripples in the leaf. You have to look at the edges of it, the margin, and there are no teeth. And then here on the right-hand side is water star grass, and very similar leaf size, but there's no obvious midvein. And the flower is very different. This flower on this one is very showy. So, A or B, which plant is curly leaf pondweed? I see the bees rolling in. That's right. So we have another pondweed here. This one's called Rabinzii, which is fern pondweed. And we have alternate leaf arrangement and kind of ribbon-like leaves, but no teeth, where this one has that obvious teeth and really nice lasagna noodle wave. Our next species, starry stonewort or Nyctalopsis obtusa. By the way, what I'm saying here is the starry stonewort is the common name. Nyctalopsis obtusa is the scientific name. This is not a plant at all, but a macroalgae. And so really what that's saying is a large algae. It looks like a plant, but there are lots of physiological differences here with the lack of vascular tissue or different vascular tissue and then others. The best way to identify this plant, because it's in the family of plants that include Chara, and Chara, also known as musk grass, and they're ubiquitous across the state and wonderful plants to have. But the one thing that separates them, that makes it easy, is the presence of these star-shaped bulbils. So this is one way for starry stonewort to asexually reproduce, is with these. So if one of these get into your lake, a new plant can grow, and it's shaped like a star, so that's where it gets its name. They are attached to this filament that looks like fishing line, very clear about that thickness. And so if you look along if you see a plant with this kind of filament and then look at it to see these stars, you have starry stonewort. Now that's not always possible where we see one, but we might see some other characters, one of them being a very smooth stem that's brittle. So between the nodes, so between here and here, is one cell. And so if you squeeze it, it pops. You can feel it kind of break and pop in your fingers, and then all of the cell's contents come out. You can take a huge mass of it, like if it's up on your anchor, and you can crush it down into something that fits into the palm of your hand. Another way to look, another clue to look for is uneven branching or uneven branchlets. And here's an example of that. So here is one of its branchlets, and there's this uneven, it's not a perfect wishbone. Instead, this one is shorter, and that's a good clue on seeing starry stonewort. Here's a picture of a real life bulbil and the filament there called a rhizoid. And then here's an example of pulling up a big chunk, a big wad of starry stonewort and seeing a star here, there's a perfect star, and those. As I mentioned, common lookalike is the native musk grass or Chara. And so musk grass is its common name because many of the species have a strong lake odor. Like if anyone's gone swimming in a lake and walked through here or lifted it up on an anchor, it smells fairly skunky, and that's where it gets its common name, musk grass. There are no star-shaped bulbils on Chara. It is a macroalgae, but it's kind of a shorter, not always, but a bit shorter in branching. So instead of starry reaching out this far, imagine it Chara only reaching out this far. And because of the cells and calcium carbonate, it can feel quite rough compared to starry. There's one more lookalike I want to mention because I've seen them in deeper waters cohabitating, is Nutella. I don't know if there is a common name to Nutella. This is just another group of species in this family. And these will have a smooth stem as well, like starry, but have even branching at the end. So at the ends, instead of that uneven branching, it has even. So which one is starry stonewort, A or B? Yes, this one is pretty obvious, right, with that star. But what we wanted to show here is that some Chara also produce bulbils, so on clear threaded rhizoids, but they are spherical, they're round. And so there you can see the difference between that and that. Our next species, Hydrilla, again, it is a prolific invader, one of the worst aquatic invasive plants in the world, and it has been invaded into North America for quite some time, but this is the first detection as of last year in Michigan. We do not want this species to become widespread because it is prolific. So how can we tell Hydrilla? We look at that the leaves are whorled around the stem, and there are always more than 4 leaves per node. And so, for example, you have 1, 2, 3, 4, 5 leaves. You look at, without any magnification, you can see the teeth on the sides of the leaves. You can see there's the real life image as well. It gives it like a really robust feel to it. Some populations of Hydrilla, and it is a common population type, is that you'll see teeth on the back side of the leaf on the midvein. The population that is found in Michigan does not have those though, but it does have the teeth on the edge. So we have a very, very common look-alike called Elodea, or waterweed. And here's the depiction showing that. So again, here is Hydrilla over here. Here are the teeth on the edge. Here's the teeth on the backside. Also, we have 1, 2, 3, 4, 5 leaves per whorl there. And our native Elodea, or waterweed, almost always has 3 leaves per whorl. 1, 2, 3. They do have teeth as well, but you would need magnification to see it. So it's not as much. Sometimes, so although almost always 3, sometimes it'll have 2, and sometimes it can have, I think I've seen 6. But when you look up and down the stem of the plant, you'll see that it very much is mostly 3, and that there's only a couple nodes that might have more than 3. So just to hammer that point home, this is not Hydrilla. This is our native Elodea. There are 2 species. There's Canada waterweed, so Elodea canadensis. And then, I don't know what the common name for this, there's Elodea natalii, which has thinner leaves than the Canada waterweed. But they both have mostly 3 leaves per whorl. So A or B, which plant is Hydrilla?
I just want to note that Joe and Christy are like super quick on these IDs. Like they always get them right away. So you guys
Nice.
are sitting on the button right there.
Joe, we need to add more of these. All right, A. Good job, everybody. You are right. This is actually a naiad over here. We had to throw in a plant, we haven't seen yet. And yes, this is Hydrilla. As you can see, what is on here, there's like these really jagged teeth, and not just on the leaves, it's also on the stems of this one, and also not whorled around the stem. There's some other kind of leaf arrangement there. And European frogbit. Hydrocharis, as this picture showed there, or as this map shows, there was a lot of occurrences of this plant along our coastal wetlands, and then has more recently popped into our inland water bodies. And this has been found in Green Bay as of last year, I believe, or 2 years ago. 2 years ago, I believe. And so that's very close to our Western UP water bodies. So please take a look. Sometimes some of our invasive species may seem far away in the UP, but not this one. This plant is a free-floating species. So we've been talking, all of our other plants are submersed, other than maybe a flower coming above, but this one is free floating. So the leaves are floating on the surface and the roots dangle below. Now, because it's a free floating species, it can be washed up onto a mud flat and appear rooted, but that is because it's been pushed onto a mud flat. These plants get pushed along in the wind and will usually grow in cattails or phragmites or just up against the emergent plants alongside of a wetland or lake, and then start to send out runners to create other plants. It's almost like, think of strawberries, how they send out a runner and then a new strawberry plant grows. That's what happens here. And it can create these really dense mats. So it's a free-floating plant. All the leaves come into one center part, so it's considered a rosette. The roots hang below, and the leaves look like little water lilies. But I don't know about you. Has anyone on this call, and you don't need to belt out, has anyone tried to pull up a white water lily or a yellow water lily? So lily pads in a lake, they are very difficult. They are firmly rooted to the sediment. So our native species are rooted. That's one major comparison. Also, these at its max width are 2 to 3 inches. Now they can be much smaller, especially when they're young, but the biggest these get is 2 to 3 inches across. And our white and yellow water lilies can be pretty small early on, but they grow much larger. And then just like our white water lily, this also has a white flower, but the white water lily, if you can picture it in your mind, has tons of petals, and it makes it very beautiful, where this has 3 petals, still pretty, but not like our yellow water lily. Here's just a picture of it growing densely. And then here is standing on a boat looking at a shoreline. So we have our narrowleaf cattail here. We have our native white water lily here. And then pushed up against, this is European frogbit. So here's even these here, those are small white water lilies rooted to the bottom. They look like Pac-Man, if you remember that. And then here is European frogbit. Which plant is European frogbit? And may I add, why? So which plant is European frogbit and why? Three-petal flower. Yep, I like it. A, looking good. So it is A. Yeah, those rounded edges, nice, Heather. Curved leaves, more heart-shaped, Kate, nice, yep. And Bill, great observation, leaf size. So if we look at our rulers on the side, and if you are signed up for the Exotic Aquatic Plant Watch, you will receive one of these in the mail, which is a laminated sheet of paper with these with rulers, so we can get for scale. And on the other side, we have tips on identifying these 5 species. But you can see this is very large compared to the max 3-inch leaf of European frogbit. So those are the species that we have you all focus on in the Exotic Aquatic Plant Watch. They are of high priority, some of them being in Michigan for a long time, but still not everywhere, of course, but in a lot of places, and then some that are more rare. Now, there are some yet where different risk assessments and working with managers around the area, we have identified, or the field of conservation has identified, other species that are of high threat. So we call those our watch list species. Some of these species have never been detected in the wild, or if they have been detected in Michigan, they're very limited distribution, might be in only 4 locations, for example. But these species have been identified to have very high potential for negative impacts. And so early detection and quick response is important. We like just to show this. If you go to this michigan.gov invasives webpage, you can find out all about these different species. We can also supply this information if you would like it. Some of these species, again, have never been found in Michigan, but but are close, and some of them have been found here already. So moving on, any questions about ID? Oh, thank you, Joe, for putting that web page up. Are there any questions before we move on to our Plant Watch protocol? Okay, well, thank you very much. If you are enjoying this plant ID training for aquatic invasive plants, Joe and I will every once in a while do plant ID training for native plants. And so you can keep in touch, and we'll always let our volunteers know when opportunities like that come up.
All right. Well, I'm going to take over from here. Thank you so much, Eric. If folks have other questions about plant identification, stuff that Eric just went over, feel free to continue to post questions in the chat. Otherwise, we're going to move on and actually get into the protocol. So let's say you decided, yep, this is the program for me. I want to be an Exotic Aquatic Plant Watch volunteer. We love that. And we also want to mention that this parameter and Score the Shore, the shoreline assessment parameter that we trained on earlier this afternoon, you can sign up to add this program to your lakes monitoring at any time during the summer through the end of August. So if you decided today that you wanted to do it, or you think about it over the weekend, you're like, yeah, this is something I want to do, even if you've already enrolled in other parameters like SecuDisc and total phosphorus, you can add the Exotic Plant Watch or Score the Shore at any time this summer through August 31st. All right, so if you were planning to do the Exotic Aquatic Plant Watch, you need to have some materials, just like always. All of this paperwork can be found on our website. We also will mail you a packet if you're signed up. That will include the data form, the instructions for building a sampling rake, which I'll talk about in a minute, a copy of that identification guide that we've already talked about, the Michigan Boaters Guide, that laminated card to take pictures of photos on that Eric mentioned, pictures of plants on that Eric mentioned earlier, a copy of the plant watch list that he showed, and a brochure about the program so you can talk about it with other folks. Additional equipment that you'll need to provide includes, of course, your boating safety equipment. You'll want an anchor because you want to be able to stay put while you're surveying an area for plants. Any plant ID guides that you like to use. It helps to have a depth map of your lake. A GPS unit or a phone that can get you coordinates is helpful for recording the locations of where you're sampling, but it's not necessary. Camera is really important. We're going to get into photographs here in a few. A fish finder, a depth finder, or a weighted sounding line can help you know what depth you're sampling at, which is useful for tracking where you're finding things. The sampling rake attached to a nice long rope. I'll show you that in the next slide. Ziploc bags and a marker for labeling them. This is really helpful because while we're sitting here at our desks looking at really nice images of the plants, it may feel like, yeah, this is pretty easy. I can identify these. But when you're out in the lake and you've got plants and they're wet and they're tangled up together and they may be tangled with some other plants, sometimes it gets a little tough to identify them on the boat. So having some Ziplocs that you can label where you found the plants to take back to shore and sit down with good lighting and you're not rocking back and forth on a boat, maybe have a hand lens or something to look at some of the features up close. That can make ID a lot easier. So have some Ziplocs in case you want to save any of your plants. Trash bags are good to have in case you pull up any trash or you pull up invasive species. Most people don't want to throw those back in their lake, so having trash bags on board will allow you to dispose of those properly. Have a clipboard with all your data forms and note paper on it, a copy of the procedures so you don't forget what you're supposed to be doing while you're out there. And of course, as we say with every parameter, a pencil or an indelible ink pen that won't run if your data form gets wet. All right, Here is the very high-tech sampling device that we use to collect aquatic plants. And when I say we, I really mean we. This is our go-to tool. We've built several of them for ourselves, for our professional aquatic plant work that we do. But you can build the same thing for yourself. These instructions, again, we can provide them to you. You want to cut the handles off of 2 garden rakes, bolt the rakes back to back with C-bolts, or I've even seen zip ties used very effectively if they're sturdy ones. And then put an eye bolt into one of the handles that you can put a nice rope on, at least 20 feet long, probably longer, especially if you have a deeper lake. This is a really effective tool. As you can imagine, dragging this along the bottom of a lake will collect a whole lot of plants. So this is your collecting tool that you'll need to construct. When to sample. We recommend sampling in mid-June through August. Northern lakes may want to begin a little bit later. Sometimes this year probably isn't as big of a deal because things are warming up pretty quickly, but you want the plants to be big enough that you can actually identify them. If you're out raking and all you're getting is tiny little sprouts, that can be hard to know what you're looking at. You don't have to do it all at once. You can survey your whole lake twice during a summer, or you can spread out your effort over the summer. Curly leaf pondweed in particular is an early season grower, and so it is likely gone from most lakes by August, especially if you live in the southern part of the state. It will go dormant by the end of the summer, and so if you wait too long to sample, you might not find it. So I wouldn't wait too long, and in particular, if curly leaf pondweed is a plant of concern to you, if you know that it's been heard of or suspected in your lake, you might want to sample early, or at least do a little bit of sampling early, just to make sure you catch that. Otherwise, I have some lakes that sample twice a year, and when they go out in August, they do not see it, even though it's there every year in June. Where should you go? Well, the first thing we suggest is you get a map of your lake. Spread it out on the table, take a look, or look on your computer at a map of your lake, and look for some important areas. Areas will be places where invasive species are most likely to appear in your lake, like boat ramps. The number one way we feel that invasive plants are getting into our lakes is that they're being moved on trailered boats that come from an infested lake and then unknowingly bring them into your lake. So look for your boat ramps, locate where they are, locate any public beaches or parks for the same reason. Any attached inlets like streams, creeks, or canals that flow into your lake can be a source of invaders from upstream. So mark on the map where those are. And also look for quiet bays and coves, areas where plant life tends to thrive. And then those would be kind of your higher priority places to look. You may then find that there's other parts on your lake that you missed a big section of lake. Maybe this whole stretch on Gull Lake didn't have any of those features. If you have the time and wherewithal, I would say add some sampling sites along there. So here's another example. It's a different lake, but what they've done is the same thing. They put a transect, which is a sampling area, by both boat launches here. And then, here's an inlet over here, and one here, and one here. Those are all the red kind of high risk areas. And then in purple, we added a few more sampling areas in areas on the lake in between those high risk areas. Now, remember, this is important. Even some data is useful. It's okay if you don't have the time or the energy to cover your entire lake with transects, even to do this many transects. If all you can do is go out and sample around your boat launch this summer, that's great. And we'd love to see that report. Just when you report, show where you sampled. So we know, and future people who look at your results know that, oh, we didn't cover the whole lake that year, but we did look around the boat launch. That'll be useful for you and your lake association or lake community. It will also be useful for our purposes. So any data is useful that you can gather. Now, you say you're ready to go out, you're heading out to sample. So we are going to describe kind of the Cadillac method of sampling. If you can do this, it's great. If you don't, again, any early detection sampling is helpful. So while we talk about a transect, what we're talking about is an imaginary line going from shore out into water, so perpendicular to shore. And this one is right next to a boat launch. You can see that this is a boat launch right here. And we set up our imaginary transect right here next to it. And we find that aquatic plants, different species grow at different depths. If you spend any time at your lake looking at plants, you've probably noticed that too. So it's great to sample at a few different depths along your transect. In this example, we sampled at about 3 feet, then we motored out to about 8 feet and sampled again, and then we went out to about 15 feet and we sampled again. And that tends to cover all the different plant communities. You may adjust the actual depths based on the shape of your lake and where you see plants growing, but this is a good starting point to give you an idea of what we're talking about. And then what you do is you actually toss the rake into the water and drag it along the bottom to pick up plants. You will want to toss it out, maybe not as far as this volunteer has managed to do, but toss it out, a nice underhand toss out into the water. Keep a hold of that rope so you don't lose the rake and the rope together. You want to keep a hold of your rope. Once it hits the bottom, drag it back up to your boat, and then you will remove and identify any plants that come up. The data sheet is where you'll record what you found. So this is the front of the data sheet, page 1. At the top, you enter your lake name and county, township, lake sampling site number that we give you, the names of all the volunteers that helped out, the dates of the survey. If it takes you several days to do it, that's totally fine. And it can be throughout the summer, just record them here. You don't have to record the time that you did it. I think we've taken that off the data sheet because it doesn't really matter. You can record any comments like unusual conditions when you were out on the water. Maybe the lake was sprayed with herbicide a week or so before you actually got out to do it. That's not the best time to do a plant survey, but sometimes we can't control when we're available and when herbicide treatments happen. So make a note if that is the case. On the bottom half of the page 1, you have a box to check that if you surveyed your lake and you didn't find any invasive plants, just check that box. And then you'll document where you actually looked, but didn't find any on page 2, which I'll show you in a minute. If you do find any invasive plants, you check the species that you found: Eurasian milfoil, curly-leaf pondweed, hydrilla, starry stonewort, European frogbit, and there is a place for other. If you find any other aquatic plants that are on the watch list or that you just want to make a note of, feel free to add those there. And then remember with your report, this isn't all that is to it. This is just page 1. There's also a page 2, which I'll show you in a minute. You'll want to include a copy of your lake map with the numbered locations of where you surveyed and any photographs taken of collected plants. And we'll talk about the importance of pictures here in a minute. I want to also emphasize the importance of this little box right here. In the past, we've had volunteers sign up for this program and they've gone out on their lake and they've surveyed a bunch of sites. And at the end of the day, they were like, we didn't find any invasive plants at all. And since they didn't find any, they never sent in their report. They thought we didn't want to know unless they found invasive species. And that's not true. We want your data, even if you didn't find any invasive species. That's data too. It's really good data. It's good news. We love seeing that. And we love having that in the record because think about this. Maybe next year or a couple years from now, someone goes on your lake. Maybe it's you, maybe it's somebody else, and they do find invasive species. They'll be able to go back to this year's report where you didn't find any and make that comparison and realize that the invasion they found may be a relatively recent one. So please be sure to let us know even if you don't find any invasive plants. Page 2 out of your 2-page report. Page 2 is where you actually put the data of where you sampled and what you found. So we recommend starting your first site as site number 1, and then you record either the latitude and longitude, or if you don't have the capacity to get that, describe in writing the location. We discourage you from using the names of people like Jim Smith's house because we have to take all that out for privacy reasons. So describe where you are in words. Road names, that kind of thing can be really helpful, landmarks, if you can't get the coordinates. List any invasive species you found in this transect. Here we used abbreviations for curly leaf pondweed, Eurasian water milfoil, or you would write none. If you took any photos, yes or no, and then any notes that you have about what you saw there. In this case, the volunteer wrote sparse because there was those 2 invasive species, but just a couple little sprigs of both. Then they moved on to their second site. Here's the coordinates. They didn't find any, so they didn't take any pictures. And you would continue down for all the sites you monitor on your lake. Taking pictures is really helpful. And in fact, we've moved to requiring pictures. So you need to include at minimum 1 representative photo of each invasive species found in your lake. And we require this so we can double check for you. It's great to have professional confirmation of what you found in your lake. So take at least 1 picture of any invasive species that you found. You are welcome to include pictures of other plants, whether you're confident in what they are or not. We can help. If you're not sure what they are, you can send them to us before you turn in your report, and we can help you identify them that way. Or you can just include them with your report. You'll want to label them. In this case, these laminated sheets that we give you are really nice because you can write on them with a dry erase marker and then wipe it off for the next plant. In this case, this was just used to identify Eurasian water milfoil, but you could also, if you were at site 2, you could write a nice number 2 here on the sheet. And that way, right there in the photo, you had location information as well. You want to make sure that your photos are clear, not fuzzy, and they should show the identifying characteristics. So in this case, we clipped off a whorl of leaves so you could easily see by the arrangement and the number of leaflets that it is truly Eurasian water milfoil. These are really good resources to have too. So, when you get that inevitable phone call from a neighbor that says, "Oh, I've got these crazy weeds growing off of my deck. Can you help me figure out what they are?" You can show them pictures of plants that you already have seen and help them identify what's growing by their deck. Again, examples, these are pictures taken by actual volunteers, just like you, of plants that they found. They took a picture, here's curly leaf pondweed, and we were easily able to verify for them that that's what it was. We could see that leaf shape and the leaf arrangement. We could easily tell what size the plant was. And then here's a picture they sent in of what they believed was Elodea, which is great. Again, they clipped off a whorl of leaves so we could see there was 3. When we zoomed in, we could see that there were no teeth on there. So it was really nice for them to have this record of a plant that looked like hydrilla, but it was the native look-alike. So it's a good idea to take pictures of natives that might be confused with invasives as well. And you can include those with your report. If you find yourself out on the lake and you don't have your handy laminated sheet with you, it's good to put something in the picture for scale. In this case, your hand works really well. It lets us know, and it lets future people looking at the pictures know how big this plant was, and that helps a lot with identification to know whether you're dealing with something really tiny or something quite large. So something for scale if you don't have the sheet. Mapping, that's the last piece that you need. So we have the cover sheet, page 2, that has the locations and what you found, pictures of any invasives or other plants you want to share, and then actually your map that shows where you went. So it can be as simple as this, where they drew on a paper map with pencil where their locations were and numbered them, and then those are the numbers they used on their report. Or you can get fancy and use Google Maps or other technology to create maps. This is, again, another real example provided by a volunteer that was comfortable using Google Maps, and she created all of these pins where all of her sampling sites were on Stony Lake here in Oceana County. And then she even made it so if we clicked on the name of the lake, it would show what species that she found. You can see that here. Curly leaf pondweed was abundant on July 3rd when she went and sampled. So we are flexible with you on how you want to do a map. You can do a low tech on a paper map, or you can go high tech with something like Google Maps. Regardless, we want your data. So submitting your data, just like all the parameters, make copies of everything. So you have a copy. Don't send us your original. That happens every year. We get a couple folks calling us because they realize they sent us their only copy of the work that they did. Enter your data into our website by October 31st, and also send a copy to us by October 31st. And for the Exotic Aquatic Plant Watch, that means the completed data form, both pages 1 and 2, your map, and your pictures. All right, a couple tips and tricks for success. Teamwork. When you're out collecting plants, it's good to have somebody who's just there to drive the boat, to get you to the places where you want to go. And then have a couple of you working on actually tossing the rake, pulling the plants up, identifying what you found, somebody to take notes. Teams are much more successful in this program than others. We actually use this program as part of a research study to see what do volunteers need to be successful. And one of those things was helpers, friends, other volunteers, neighbors, other people to come out and help them with it. When you try and do something like this by yourself, most people might get a little worn out and tired of it before you get done. So volunteer teams are more likely to complete the sampling, submit the data, and continue in the program. So we encourage you to find some colleagues to help you out with it and have some fun with it. We also learned through our study of this program that it does help for new volunteers in the program to get a little help out in the field the first time. So we were able to apply for and receive some supplemental funding from the state of Michigan through Eagle from the sale of the water quality license plates here in Michigan to help us actually come out and provide field visits, so on-site visits for some of our new volunteers in this program every year. So we may be able to visit your lake to help kick off your survey, to assist you with plant ID, and answer any questions that you have. So you're really well prepared to be successful with the Exotic Plant Watch. We'll be arranging those in the next few weeks from the volunteer lakes that are enrolled so far. We can't get everywhere, and new lakes are our top priority. So if you're a new lake in the Exotic Plant Watch and would like a visit from myself or Eric or someone else from our team, let us know, because we'll be planning those fairly soon. All right, that was the end of my slide. So I'm going to say, hey, do we have any questions? And I think I'm going to stop sharing my screen at this point.
We don't have any extra questions, so please, everyone, write that. Mary is telling us that they would love a visit to for us to visit Grand Lake over in, I think, Alpena area.
All right, that sounds good. If you're signed up for the program, you are on our list, and we'll remember that you made that request.
Joe, if you, I answered a bit about this, but just in case you have some extra points, how long after monthly herbicide treatments is the best to do a sampling for the Plant Watch?
Yeah, that's hard, right? Because it sometimes takes a while for those herbicides to show impact. But yeah, I mean, I would say I would avoid going out immediately after in the next couple weeks. But I would definitely, but what's most important, I think, is to make a note about when those treatments happen, like in terms of dates, and have that for context in your report, because that may definitely influence what you see. And you can also make notes about the condition of any plants that you collect. Like if you say, yeah, I found Eurasian water milfoil, but it all looked like it was dying, make a note about that and the fact that there was an herbicide treatment.
Are there any questions, any more questions about the Exotic Aquatic Plant Watch or any other parameter that you saw today?
We are happy to stick around till 4:30 because that's our actual ending time. So if there's anything else, this is your chance.
If there are some folks who have not enrolled yet, I put in the link to, if you haven't enrolled yet but would like to, you can visit our Become a Volunteer page and learn a little bit more and go from there.
Well, it's been fun doing this. As I mentioned at least one point earlier today, we've been doing training for a few times this season. We're trying to offer more and more options for training. And so we have, we were at the MLSA conference last week, online this week, and then we will be in the Western UP in Crystal Falls, May 18th, doing this again. So we want to make this as accessible to everyone as we can. I saw a question in the chat from Sue about the Montana resource for genetic testing. I way earlier today during this training, I put in the chat a link to our report that we did. Eric and I wrote this with some colleagues, including Ryan Thum in Montana, about hybrid water milfoils and how to manage them. So there's an MSU extension report about that. And then I see Eric just put in a direct link to Dr. Thum's lab. Again, that's who we worked with. But yeah, if you're interested in hybrid milfoil, I would highly recommend reading our report that we wrote about it. Not to be self-promoting there, but it's a really good resource that explains what hybrid water milfoil is and the study that we did, because and we involved a lot of CLMP volunteers in that study looking at milfoils around Michigan and to see how widely distributed the hybrids were and whether they were resistant to herbicides or not. And what we find is there's a huge genetic variation and variability from lake to lake across Michigan. And some of the hybrids are herbicide resistant, others are not. So Ryan's lab is doing testing of the various varieties in the lab to see if they're herbicide resistant or not. So that's really valuable information to have if you're thinking about a treatment on your lake, to know whether your money is well spent or maybe wasted if you have a resistant strain. I see a question there. Our neighbors want to begin an herbicide treatment. Are there any that you recommend? Well, we're inching into a management question there, but we're only got a few minutes left. And this was a pretty quick one for us to answer because we have to say we don't recommend anything. What we recommend you do is you get in touch with the state of Michigan's Aquatic Nuisance Control Program. And that's just michigan.gov/ANC for aquatic nuisance control. And there you find a wealth of information about different treatment options, what they are approved for or not approved for. And that's the unit that eventually approves or disapproves permit applications to do herbicide treatments on lakes. And they are more than willing to answer your questions. Do not be afraid to call them up. They're not just an enforcement and regulatory body. They are excellent educators. They know more about the herbicide and other management options that are out there than anyone else in the state, I feel. And so they would rather have folks contact them and ask them questions than just blindly go forward. So, don't hesitate to reach out to them with questions like that. See another question. I'm curious, if I collect some plants, could I send them in to see if I am recognizing them correctly? At this time, I'm slightly hesitant to commit to my obligation since I am a newbie. Yeah, Sherry, we're happy to help you with plants. Even if you're not quite ready to sign up, if you want to send us pictures of plants that you're finding, we would be happy to take a peek at those and let you know what you're looking at.